So thank you and thanks the organizer for inviting me here. It's a pity that I was not here the previous days, but I had another meeting. I was told by other speakers and guys from here that the meeting has been very nice so far. So yes, I'm working now at the Neuroscience Institute in Pisa. But I'm still working at the European Laboratory for Nonlinear Spectroscopy. I will give a very general overview of microscopy techniques and correlative microscopy techniques to study different aspects of the brain. I would like to keep it as informal as possible, so please feel free to interrupt anytime and ask whatever question comes to your mind, okay? So, why do we need multiple techniques? Why there is no one technique that allows you to investigate whole scales of the brain? Because the brain is an extremely complex organ whose complexity arises from the different level of organization. So we have the entire brain, who's roughly organized in areas, in functional areas, the motor cortex, the visual cortex, for example, the whiskers. No, we are not mice, but all these areas are actually composed by neurons and glial cells, which form a small network, and all these small clusters of neurons are actually responsible for these local functionality. They get the input from other clusters of neurons and they process it and give an output. So this processing happens also at the single neuron level, where the dendrite collects information from a lot of other cells and neurons, and then at the somal level the signal is filtered and processed and sent to, and the relevant information is sent to other neurons. The processing happens even at the finer scale, so at the dendritic level, axonal level, but at the synaptic compartments too. So dendritic spines and these vesicles, and boutons, which I will talk about during the talk are pre- and post-synaptic compartments, which are responsible for the chemical transmission of the signal. So by the synaptic vesicle release, the relevant message is transferred from one neuron to the other. To target all these different layers, you need different techniques that goes from the magnetic resonance imaging to light sheet, two-photon, going to the super resolution techniques, and finally electron microscopy. So there's no one technique, and I will go through how you can use some of this technique to tackle these different aspects of brain structure and functionality. So let me go through a few examples of the different layers of complexity. So at the whole brain level, the so-called macro level, we have magnetic resonance imaging, which is a really important technique for mice, but mostly for humans. It gives us access to the clinical side, and it's actually quite broad, because this is an example of activated brain in set condition or in happy condition. You can roughly identify some red, so activated area. The technique is quite low resolution, but it's non-invasive, so it can actually give you important information on the human brain. All these activated areas are actually composed of neurons and cells. So here, this is like the so-called mesoscale, so the centimeter or hundred of microns level, where this organization of clusters of neurons produces the final activation of these areas. We go to the finer scale, where from the two-photon microscopy that can be used to analyze this small scale circuit, we can go to the fine details of single neurons. So you can actually anatomically depict and image the dendritic layers and reconstruct single neurons. But you can go also with two-photon microscopy to the synaptic level and define the plasticity of this small structure that protrudes from the dendrites, in this case, so spines. This is a fluorescence image of dendritic branch. All these green dots are actually synaptic structures. Two-photon is the limit where you can use this technique to investigate micron-resolution scale structures. But for spines and subcellular structures, the election techniques might be either in the optics field, a super resolution, and in the other very efficient alternative is electron microscopy. So electron microscopy allows you to see very small structures. Maybe I can use the pointer. Subcellular compartments, so you can actually, by using heavy metal staining of the cells, you can actually see membrane, myelin sheets, you can actually see synaptic vesicles, and you can distinguish the postsynaptic density. So you can actually, this is one of the only techniques that can tell you if a synapse is actually active. So if there's all the synaptic machinery is there. Why? So in my talk I will briefly give you some examples of correlative techniques we have been used so far in our lab, and then I will go through the description of a specific use case of the Human Brain Project that works on the use of different microscopy techniques to tackle a specific biological issue. So how stroke and rehabilitation works on brain plasticity. So let's start with the correlative techniques we've been using so far. And in this case, I'll show you some techniques to find some correlation at from the micron level. So you can have single cells seen through two-photon microscopy with the finer details of electron microscopy. So just a brief introduction. I've been told to be quite general, so I will give you a couple of sentences on how, on how two-photon fluorescence work. This is the absorption and emission transition for a single fluorophore. So you have the absorption of a photon of a certain wavelength, and then the emission of one photon of a shorter, usually a higher wavelength. Then with two-photon microscopy, you have the simultaneous absorption of two photons at the approximately double the wavelength allows you to obtain the same fluorescent emission. Why this process is so interesting is that this is because the nonlinearity of this interaction. So the probability of this transition is no more linearly linked to the intensity of excitation, but to the square. So this is extremely important for the reason you see here. So this is one photon excitation where all the not only the focal plane, but all the planes that are above and below here are actually excited. This produces not only photodamage, unwanted photodamage and photobleaching, but also a lot of noise coming from these other layers that you are illuminating. In this case, this is what happens with two-photon microscopy, with two-photon excitation, sorry. So here, this tiny dot here is actually the focal spot. So this transition is, let's say, so unlikely to happen that it happens only here at the focal spot. So it allows you to have intrinsic optical sectioning, and you can, by scanning the laser across the XY plane, you can actually reconstruct the image, and then by using system like the remote focusing, or more generally CADS system, you can actually scan the beam in the objective plane in Z and reconstruct 3D volumes. Another advantage of two-photon excitation is that the light that is being used is in the infrared range, so the light penetrates a lot more inside the tissue due to the less scattering of endogenous molecules inside the tissue itself. So it's actually it has a lot of advantages and allows you eventually to go to image in vivo inside the brain of a mouse. So this is an example. The only thing you need to do before starting is to put a cranial window on top. So you need to remove the bone, which is highly scattering. The near-infrared light cannot really go through the brain to allow you to have this level of resolution and penetration inside the tissue. So this is 3D, lateral reconstruction of the images of the neurons you can see with two-photon microscopy through the cranial window. So you can actually see dendrites, and you can go up to the soma layer of layer 6 neurons inside the cortex. This technique has the resolution that allows you to see single synaptic contacts. So these dots here are dendritic spines, and this here is a bouton on an axon. So this you can repeatedly image this structure over time under this cranial window and get an overview of of the dynamic of the events happening in the brain. So this is an example of what you can see. This is a flying inside the brain. This is you can see dendrites, axons. The dots here and there are spines, and you can go even to colors. So depending on the detection scheme you use, you can actually see with the same resolution different colors in the same image. I would like to go through the correlative study we've done so far. So by using, why do you need a correlative approach? In our case, we wanted to be certain that the formation of new synaptic contact corresponded to actual active contacts. So we use a combination of laser exotomy. So we use laser to actually produce in vivo a small ablation on single axons. So we produce this is a time lapse of what happened by focusing the laser on a single vesicle, on a single spot on this axon. And after seeing the degeneration of this some tens of microns of an axonal structure, we surprisingly saw some formation of new branches from the same axon. So you know quite well that the central nervous system is quite in a mammalian is quite hard to. It's quite hard to find some spontaneous regeneration inside the central nervous system of mammalian animals. So we found it quite surprising. So we wanted to investigate if these branches were actually underlying some real synaptic contacts. So we contacted the group of Graham knot at EPFL, and we say yes. So we want to find this structure that we've seen so far with two-photon microscopy. This was the situation before this so-called laser exotomy. After that, we found this, after some days on the same structure, we found the formation of this new structure. So are these dots here corresponding to synaptic contacts? This is quite tricky, I have to say, because it's like, given the very different resolution of the two techniques, it's really like to find a needle in the hay, because you cannot also find the fluorescence of the structure is lost. So it was quite complicated. Given also that the main technique that was used since, I would say, some tens of years ago, the most commonly used technique was transmission electron microscopy. In this case, the correlation was done by taking pictures of the cranial window. So this dot here was where the neuron of interest was. Then you slice the sample, you resin embed the sample, you slice it, you try to reconstruct by overlaying all the tissue layers where was your neuron of interest, and then you go to this very time-consuming slicing of the sample. So this it's not only the time-consuming part is not only the production of these very thin slices cut by the diamond knife, but then you have to image them and find and reconstruct the 3D structure and identify the neuron you were looking at. So this could take like the whole PhD thesis of one guy who could be just simply lost in this reconstruction and who could have this PhD completely fail, let's say. So then this, I know guys where it actually happened. So eventually focused ion beam milling came to help. So this technique combines serial electron microscopy with the gallium beam that actually burns the first layer of the tissue which has been already imaged. So you image the surface, then you burn the first layer, then you image the following layer, and so on and so forth. So in this case, the sample is fixed, so it stays there, and you just go through the volume you need to image. So that helps a lot. Another trick that we used was to use the laser to actually burn a laser box next to the axon we wanted to find, so that this laser burn can eventually be found in the resin embedded sample, and that helps a lot to find the needle. So this is the 3D reconstruction we got by using this combination of techniques. We could find this exact axon, and we could actually see if this button was forming synaptic contacts by verifying the presence of synaptic vesicle and all the synaptic machinery inside the axon. I show you an example of going from micro to nano. Then for other reasons, we are also interested in going in the other direction. So from the information that you get from two-photon microscopy, for example, which is really useful in terms of dynamic events. So with two-photon microscopy, you can visualize the same structure or the same neuron over and over again under the cranial window, so you can get a hint of the dynamic that happens inside the brain. But the penetration of the technique is such that you cannot achieve more than, I would say, less than a millimeter inside the brain. So you can access only the cortex and not more. If you want to see what this neuron, who this neuron was contacting and contextualize it, you need other techniques like light sheet microscopy. So this technique is basically it's based on the use of a sheet of light that goes through the sample, and the detection of fluorescence is orthogonal to the image plane. So this technique combined with cleaving agents allows you to perform a very fast scanning of the entire brain and reconstruct the anatomy. Very fast meaning that the imaging time for one mouse brain is 1 to 3 days, let's say. This technique is non-destructive. The only problem is I would say, it's that it doesn't allow you to, this is done on fixed brain. So it's nice to have a combination of light sheet microscopy and two-photon, because here you have the whole scale, you have the synaptic details over the whole brain in the fixed mouse, but you don't get the dynamic. The dynamic, you can get it with two-photon microscopy. So the system that we have in our lab is I won't go through the details. This work done by Ludovico Silvestri. He made this confocalized light sheet microscope. So it basically have a special filter, a slit here, that filters out the out of focus photons. So you can fastly get a 3D image of the sample with a higher signal to noise ratio. This is some of the images that you can get from from light sheet microscopy. This is work done also by Mary from Caroline in our group. You can have like the whole brain reconstruction of brains which have been either transgenically expressing fluorescent protein, like this is an example of parvalbumin GFP labeled neurons. This is GAD, but you can also use dyes. So this is propidium iodide, and this is the reconstruction of the column of the brain, and you can also reconstruct blood vessel. I will show you some nice results we have on blood vessel at the end of the talk. And the
same sample?
No. These are different samples. So these two are two different transgenic mice. This is some labeling with PI, and this is another type of labeling on another mouse. Yeah. And this is roughly the resolution you can get. So you can see single axon, you can see, of course, single cell, and the finest capillaries can be dissected. So this is the example of correlative light, two-photon, and light sheet microscopy. So here was the field of view of the image collected by aligning different stacks on a live brain. So this is done in vivo. You can get the details of the synaptic contacts, and you can follow the dynamic over time of these structures over time. Then, in order to retrieve the same structure, you need to follow some quite tedious procedure, I would say. You have to fix the brain. Once you have, once the time series, so the longitudinal experiments, is finished, you take the brain, you perfuse it, you put it in clearing agents. We recently developed a versatile clearing agent for fast clearing of the brain, and then you image with light sheet microscopy, trying to find the same region. This is the procedure that we used in this case for clearing the brain. This is the final product, so you can see that the lines below can be actually seen, so the brain becomes actually more transparent. This is not so recent work. We now move to clarity and other clearing agents which produce also less shrinkage of the brain, but you basically have to wait and change solution and wait. That's the procedure. And in order to retrieve the same structure we were looking with two-photon microscopy, we took advantage of these black shadows which actually represent blood vessels. So here is the two-photon image. This is the reflectance image. So you see the contrast of the blood vessels in this image are given by the absorption of hemoglobin inside the brain by using green light excitation, and you can actually retrieve also by black shadow the blood vessel we were looking at in the cleared sample imaged by light sheet microscopy. So this is the volume rendering of the block we've been imaging so far with light sheet microscopy, and this is two-photon. And we can actually see by some of the features of these neurons that actually the neuron we found on confocal light sheet microscopy is the same, and you can actually then reconstruct this structure and see who this neuron is contacting and contextualize it in the wider framework of this microcircuitry. Another nice technique that has been developed so far in our lab and that exploits light sheet microscopy, this is done by Ludovico Silvestri, is the use of the ex vivo investigation of neuronal activity. So by using CFOS mice, he could, in these transgenic mice, the early gene expression is associated with the expression of GFP, so you can actually see through their fluorescence whose neuron has been activated by a specific behavioral task. So you can have this, you take this neuron, you have the, by imaging with light sheet microscopy, you can retrieve and define the position and the type of cells which have been activated over time by, as I told you, a specific behavioral paradigm. So we also use semantic decomposition algorithms to find the position, to determine the position of the neurons inside the whole brain. This is actually half a brain, but it's now a work in progress. We are reconstructing the entire activated brain inside a mouse with light sheet microscopy. I will now go through, so in HPP, I'm involved with Francesco Pavone in SP1 for the experimental part. So we are deeply involved in the co-design project, where the research use case is on the mouse brain plasticity after stroke and after rehabilitation. I'm also scientific coordinator of SP10, where we try to simulate this experiment in the Neurorobotic platform. And in SP1, we use a combination of optical imaging techniques to investigate, to address different aspects of brain functionality. What I've shown you so far was mainly on the anatomy and on the morphology of neurons and cells. Now, what we are interested now in the more recent, in our more recent experience, is to investigate the functionality. So, why we are interested in stroke? Stroke is a devastating disorder which affects 15 million people worldwide. That's a really big number. It's every year, and for the survivors, which are only a fraction of the affected people, it causes long-term disabilities. There are two main modalities of stroke. One is the hemorrhagic stroke, where the blood vessel leaks out blood, and the formation of a ischemic stroke, so a clot inside the blood vessel that prevents the blood flow and eventually leads to the degeneration of the tissue, of neurons and the tissue. What I will talk today mainly is the focal cortical stroke, so where the stroke is focused in a specific region. This is at the cortical level. This leads persistent motor deficits, which can, to some degree, be recovered spontaneously. So when I talk about stroke and degeneration and spontaneous plasticity, what do I talk about? So this is the pre-stroke condition. So you have in this picture the cortex, subcortical layers, all these blue and red cells are neurons. They are connected within the cortex. So within the same cortex, these neurons are connected. They are connected also with subcortical layers, and through the corpus callosum, mainly they are connected with the other hemisphere. So when a stroke happens, the blood flow decrease in that region leads to some metabolic dysfunction that affects not only the injured region, the oxygen-deprived region, but also all the regions that are connected to it. So, for example, this, which is called the peri-infarct region, so all the neurons which are actually connected here with the injured part, the subcortical region, and the contralateral area. So this type of injury affects, but as many others, it affects the connectivity at the regional level, at the mesoscale level, also, more than the remodeling of blood vessels. So something about what happens afterwards naturally is shown here. So you have the formation of new patterns of connectivity of the rescued cell, of the spared cells, with the subcortical neuron and contralateral side. This happens, has been reported mainly by the group of Carmichael, that is happening through the sprouting of new branches, but also involves at the synaptic level the remodeling of the circuitry in terms of contacts. So this is an example of what happens of the different type of neurons in the pre-stroke condition. All these dots here are synaptic contacts. What happens in the stroke condition is that these neurons are deprived of synaptic contacts because of the death of these neuronal cells. And what happens a few weeks after stroke, in the so-called post-acute phase, is that the new contacts are formed and a new excitatory-inhibitory balance is achieved. So there are some degrees of spontaneous reinnervation, somewhat the activity is restored, but the overall picture is that the motor deficits are still heavily burdening the patients. So there are a lot of efforts to stimulate the cortical rewiring after stroke. This is done mainly by rehabilitation, so motor training of the injured limb. This actually is thought to promote functional recovery. The mechanism by which this happens is not very clear yet. What we are mainly interested in is robotic rehabilitation. So by the use of a robotic device, you can actually produce a non-invasive, accurate, highly repeatable, and it's an instrument where you can actually measure and quantify the performances and the recovery of the patient over time. So some studies, basically ex vivo studies, show that sprouting is happening naturally after stroke, and they've shown so far that rehabilitation might actually work in this direction of promoting, further promoting sprouting and reinnervation of the perilesional area after stroke. Other studies suggest that actually the rehabilitation might promote the stabilization of the new, not just promote the formation of new contacts, but stabilize the new functionally active circuits. What we've been using so far, we've been developing a robotic platform for the rehabilitation of the mouse. So this device is quite similar to what has been used in humans, and it's quite easily performable by the mouse. So this is the head fixed mouse. We needed to record this activity. We need the head fixation because we need to record the activity, what's happening inside the brain while it's doing the task. Then we have this, we motivate the mouse to do this task by using condensed milk. So every time he does this pulling task, he actually gets milk reward. Then what he has to do is rather simple. So he only has to, when the motor extends his forelimb, he has to actively pull it. So that's quite simple for the mouse because he just needs to go back to his natural position. He does this task some kind of 10 minutes every day for a month. That's his rehabilitation task. What we measured simultaneously with the cortical activity is also the forces that the mouse applies. So here there's a load cell which measure the forces that the mouse applies in the 3 direction, and we also measured by using 2 LEDs the position of the forelimb over time during the task. This has been done in collaboration with Professor Micera at EPFL, who developed the platform, and Dr. Caleo, who worked in the Neuroscience Institute in Pisa. So for the experimental paradigm, we don't wait mice to have a stroke naturally. It takes some time. So we use a photothrombotic stroke model. We use light. We are kind of too much focused on light, so we use light even to produce a stroke. We, by injecting photosensitive dyes called rose bengal, we can actually produce the formation of blood clots and dye clots inside the blood vessel, and therefore block the blood flow. This is an example of a single blood vessel occlusion. So this is the beam of light. You can progressively clot permanently the blood vessel and prevent the blood flow. What we do is usually extend this blood flow deprivation and blockage over the entire motor cortex. So we want to disrupt the activity of the primary motor cortex. So this is a region of about 1 millimeter in diameter. What you see is that the blood vessel pattern, as seen from the cranial windows, change quite consistently from here, which all the blood vessels are very linear, and here where you have like a very huge rearrangement of blood vessels. This is the coronal section to show that the actual lesion is performed and affects only the primary motor cortex. So this, where this dip is where we perform the lesion, which corresponds to the primary motor cortex. So this method is quite reproducible and targetable to the regions you're interested in. For the rehabilitation paradigm, we use basically a month of rehabilitation. So this mouse just does this pulling task every day for 5 minutes, starting from 5 days after stroke. Why we need multiple level investigation? Why we need multiple optical techniques to target rehabilitation in this plasticity? Because as I told you previously in the presentation, there are different levels of complexity and organization which are affected by stroke and then by rehabilitation. So the blood vessels are on one side, one aspect. One other is the synaptic level plasticity. Another one is the remapping at the mesoscale level, and then also the interhemispheric connectivity is changed after stroke and rehabilitation. So we use we perform wide field mesoscale imaging of Taiwan GCAM6 mice. I will just give you an idea of what these mice are, why they are important, to perform a quantitative evaluation of the motor performances in parallel with the remapping of the cortex over rehabilitation period. Then we perform, by using an all-optical approach that combines optogenetics and wide field fluorescence imaging, we investigate how the interhemispheric connectivity is changed after stroke. Then we use two-photon imaging to go to the finer scale and address the plasticity at the synaptic level with two-photon microscopy. And finally, by using stereo sectioning two-photon microscopy combined with cleaving agents, we see how the vasculature is reshaped by rehabilitation. Just a few words on, I told you we have this model which is called Taiwan GCAM6 mice. What is GCAM and why is calcium important? Why do we need calcium to reveal cortical activity or neuronal activity in general? The calcium is involved maybe most of you know already, but calcium is involved in neuronal activity at many levels, at the synaptic level for the release of vesicles, of neurotransmitters. It's involved also in voltage sensitive channels. So it acts at many levels of the neuronal activity. So calcium, the variation in calcium concentration is actually very important to address, to have an idea of the activity inside the brain. What has been used so far, historically, are calcium dyes. So these basically are chelants of calcium, which upon binding of calcium become fluorescent. But the main problem of calcium dyes is that actually they are not stable over time, so you can actually image just once. If you want to perform longitudinal imaging, to have an idea of what's happening over time, you need transgenic animals and or genetically labeled cells. Therefore, we use GECI, which are the so-called genetically encoded calcium indicators. These are proteins that are that can be expressed in neurons. This is an example of this. There are now a lot of variants available. GCAMP is one of the most commonly used now, GCAMP6 especially. This is a protein which has a calmodulin module which binds calcium, and upon conformational modification, the protein becomes fluorescent. So you can actually see, by seeing the increase in fluorescence in the brain, you can see that the neurons are actually active.